So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at prokaryotic cell replication and viruses. Okay, so last lesson we looked at mitosis, and we looked at some exam questions with mitosis, and the lesson before we looked at cell fractionation, um, and we've basically come to the end of this unit. This is the last topic in this unit, and it's about prokaryotic cell replication. So we've looked at eukaryotic cell replication. We're going to have a look at prokaryotic cell replication now. If you guys don't have access to the Q&A, you can scan the QR code, which is on the screen. That QR code will take you um, to a form which you can ask questions for. So do we do OCR-A? We don't primarily do OCR-A. We normally follow the AQA structure from the AQA specification. However, I do know that a lot of the content overlaps. So you just have to sort of decide what's on your spec and what's not, basically. Okay, so if we look at what the spec is for today's lesson, this is what the specification says. We're going to start off by looking at prokaryotic cell replication. Then we're going to move on to viruses, and we're going to finish off with some exam practice. So the specification, as I stated, is the second part of the mitosis specification, and it's the last part of cell structure. So we're going to be looking at viruses, how viruses replicate, the structure of viruses, which I believe we already have looked at in previous lessons. We're going to look at binary fission because this is from your GCSE biology. And there's a bit more detail that you need to know about binary fission at A level. And then we're finally going to finish off by looking at some calculations at bacterial replication, and then exam questions at the end of the lesson. Okay, is the spec similar to Edexcel? All the specifications are very similar. There's a lot of overlap between all the specifications. There are a few topics here and there which are a bit different. However, the majority of it will be very similar across all the specifications. Okay, so if we begin now, we're going to start off by looking at prokaryotic cell replication. Now, this is basically your GCSE sort of knowledge, but a bit more sort of extended because you're meant to know the process in a bit more detail. However, there's a very big overlap. So we know prokaryotic cells, the first thing that comes to mind is bacterial cells. Bacterial cells are an example of a prokaryote. Okay, and how do prokaryotic cells replicate? Well, they replicate through binary fission. That's the process that prokaryotic cells go through in order to replicate. And you need to know the process of binary fission a little bit more. And this is what happens. So previously, we've looked at the definition for the structure of bacterial cells and what a bacterial cell contains. We know that it contains circular DNA, it contains plasmids, it contains a cytoplasm. Basically, these structures are the ones which replicate. Okay, the process that they replicate by is binary fission. Okay, so this is your process. And then the internal structures, one of which is the circular DNA and one of which is the plasmids, two key structures when it comes to DNA. They are the ones which replicate, and then the cytoplasm divides in order to form your replicated bacterial cells. Okay, so these are the four processes. And in a exam question, this came up very recently. I think it was in last year's paper, A-level paper as well, that they said describe the process of bacteria cell replication. Okay, that's what they said. And it was worth 3 marks. And the 3 marks, one was coming from stating your binary fission. Okay, one was coming from these two parts, and then one was coming from that part. So this is a mock scheme answer broken down. So I think this it's come up twice before, and last year was one of the times that they asked it. I think it was in paper three of A Level Biology. But it was the very first question on that paper, and it was literally the mock scheme was broken down exactly like this. How do bacterial cells replicate? Step one, you go through binary fission, then you replicate the circular DNA and the plasmids, and then you divide the cytoplasm divides and it forms its identical cells. Okay, and the key point that you need to know that over here, um, we know that we're going to be forming daughter cells. Okay, after we've replicated the cell, after the cell has replicated, we are going to be forming um, daughter cells, just like that. Okay, now when we form these daughter cells, if a daughter cell doesn't receive circular DNA, if it doesn't receive at least one plasmid, then the daughter cell will die. Okay, so if this over here, if the first daughter cell, if it doesn't receive a plasmid or a piece of circular DNA, then this daughter cell will literally die. And same goes over here. If the second daughter cell doesn't receive a plasmid or circular DNA, then this will die. Because without a plasmid or circular DNA, the cell can't carry out its optimal functions. Okay, so bacterial cells, it is required that they need that circular DNA and at least one copy of a plasmid. If that doesn't happen, then it will destroy itself. Okay, so that's the process that you need to know in terms of prokaryotic cell replication, and it's a very small part of the spec. That's why, and there's not many application questions that you could get towards binary fission. It's literally just an extension from GCSE. But because it's a very small part of the spec, it can be assessed as well. They can put it in because people tend to ignore it. But these are your mock scheme answer that you need to have in order to answer an exam question. Okay, so if we go back now to the specification, we can see that binary fission is in prokaryotic cells involves the replication of circular DNA and plasmids, division of the cytoplasm to produce two daughter cells, each with a single copy of circular DNA and variable copies of plasmids. So again, it's highlighting the fact that if it doesn't have at least some circular DNA and at least one copy of a plasmid, then that daughter cell will destroy itself. Okay, but the key points, again, these 2 points over here are very key, and the process of binary fission. Okay, so prokaryotic cells replicate through binary fission. They will replicate their circular DNA and then they will replicate their plasmids and then they will split off into two identical daughter cells. And each daughter cell will have circular DNA and plasmids. If either of them don't have circular DNA or plasmids, then the cell will destroy itself. Okay, so now we've looked at that. The next thing you need to know, and which could be assessed in an exam, is the point about the formula. Okay, you could be asked some calculations, and these calculations normally tend to come up in paper three of A Level Biology. It's basically where all your so whenever you've got some required practicals coming in, they like to put it in that. You've got the culturing microorganisms, a required practical, which also forms a base of this, but there's a simple formula to follow. Okay, so we know that bacterial cells, they replicate by binary fission, and they go through binary fission once every 20 minutes. If the circumstances, if the atmosphere, if the environment is favorable, they can replicate once every 20 minutes. Okay, so that's very fast. If you compare it to a normal body cells can take ages to go through one cycle of mitosis. However, bacterial cells, they replicate once every 20 minutes if the conditions are favorable. And the formula that you would use to calculate the number of cells after n generation. So what that means is basically, if I started off with one cell over here, this cell is going to go and replicate. like that. It's going to continue replicating like that, and it will continue and continue and continue on replicating. The purpose of this formula is to work out the number of cells after a number of generations. Okay, so after one generation, you form two cells. After two generations, you form four cells. And then if we keep on going, we can see that we're going to be forming more and more cells. Okay, so as you can see, we've gone from, in our first generation, we had two. In our second generation, we had four. And then in our third generation, we have eight. So as you can see, we've doubled. Okay, so in the next generation, you're going to have even more. Okay, so that's what the purpose of the formula is. The purpose of the formula is basically to look at how many um, bacterial cells are present after a number of generations. So we can say after 3 generations, there are 8 bacterial cells present. After 4 generations, there will be 16 bacterial cells present, and so on. And the way that you would do this is you would have the initial number of cells. So the question will give you there's an initial number of cells. For example, in a population, there are 5 bacterial cells. How many bacterial cells will there be after 5 generations? That's what you could be asked. And we're going to have a look at an example question as well and how to work it out. But basically, a question will always give you the initial number of cells. So this is your starting population. That's your starting population. And then you multiply it by 2 to the number of generations. Okay, so this is after how many generations, after how many rounds, how many bacterial cells are present. Okay, so for example, as I said, a very simple, if I told you that the initial number of bacterial cells in a population is 5, which is a very simple number, and then I wanted you to work out the number of bacterial cells after 10 generations. And the reason why this comes in a lot with culturing microorganisms is normally you have your petri dish and you normally culture your bacteria on your petri dish, and then you want to work out how many cells there are after this many generations. Okay, so the way that you would do this, you have your initial number 5, and then you multiply it by 2. Your base number is 2. And the reason why it's 2, if you didn't already know, is basically because one cell divides into 2 cells. Okay, that's why we have a base number of 2, because each cell divides to form 2 identical daughter cells. If that number was 4, then the base number would also be 4. Okay, but because this is dividing into 2 types of cells, that's why the base number is 2. Okay. And then 2 to the power of the number of generations. I've told you that it's 10 generations, so it's going to be 2 to the power of 10. Okay, so you would just do 5 times 2 to the power of 10. And that gives you an answer of 5,120. So that tells you that after 10 generations, there will be 5,120 bacterial cells. Okay, so that's how you use the formula. You have your initial number of cells, your starting population, you then multiply by 2 to the power of number of generations. Okay, so we have a question over here. You could also be given a question like this. The number of generations, okay, if I go back and I told you that bacterial cells replicate every 20 minutes, how many generations would occur in 1 hour? So if I said to you, bacterial cells replicate every 20 minutes, how many generations would be present in 1 hour? Okay, so we know that 1 hour has 60 minutes. Okay, there's 60 minutes in 1 hour. So that, and if one bacterial cell takes 20 minutes to replicate, in 60 minutes, you would just do 60 divided by 20, and that gives you 3. So there are 3 generations that are going to occur. That's how you work out the number of generations. You literally look at the time that it takes for one cell to replicate, and then you look at the total time that you have, and then you divide the two. So the generations in 1 hour, you first convert it to minutes, so it's 60 minutes. Then you would do 60 divided by 20. That would be 3 generations. Okay, so what I want you guys to have a go at, I want you guys to have a go at this question over here. And this is a representative question of what you could get asked in an exam as well. Okay, so what you can do is have a go at this question and let me know your answer in the Q&A, or you can let me know your answer on the form by scanning the QR code if you don't have access to the Q&A. Okay, getting a lot of correct answers. So well done, everyone. I'm going to go through the answers now. So if you have a look at this question, we know that the initial number, so we know that a culture seed contains 5 times 10 to the power of 5 cells. Okay, so this is the initial population. Okay, so we have our initial population here. Okay, that's our initial population. We know that the doubling time, of this population is 10 minutes. Okay, and we know we need to work out how many there will be after 5 hours. So the way that we would do this is step 1, we would work out the number of generations. So work out the number of generations. Okay, and to work out the number of generations, you need to look at the doubling time, which is 10 minutes. Before, in our previous example, it was 20 minutes. Over here, it's 10 minutes. And then we need to look at our total time in minutes. So the total time is 5 hours. To convert that into minutes, we just do multiply by 60, and that will convert the 5 hours into minutes. Okay, so it would be 300 minutes. So we know that there is a doubling time of 10 minutes, and we know that we need to work out the population after 5 hours. In 5 hours, there are 300 minutes. So how many generations are going to go past? You have to do 300 divided by 10, and that would get you 30. Okay, it gives you 30 generations. So that's the first step that you would do. And normally if this is worth 2 marks, that will be worth 1 mark. Okay, now in biology, maths questions are worth very few marks, even though they're very difficult. So over here, this would normally be worth 1 mark. Okay, so if you've got a doubling time of 10 minutes, after 5 hours, you do 5 times 60, gives you the minutes. If one bacteria takes 10 minutes to divide, if it's given 300 minutes, then you would just do 300 divided by 10 to work out the number of generations. So there are 30 generations that it's going to go through. Now, once you've done the generations, you then use the formula. So you use the formula, and the formula is initial population multiplied by 2 to the number of generations. Okay, so the initial population, as you can see, is over here, 5 times 10 to the power of 5. So it's going to be 5 times 10 to the power of 5. That's your initial population. And then you're going to multiply it by 2 to the power of the number of generations. Okay, and the number of generations is what we've just worked out over here. So it's going to be 2 to the power of 13. So that's what you need to type into your calculator. 5 times 10 to the power of 5 multiplied by 2 to the power of 13. And that gives you an answer of 5.37 times 10 to the power of 14. Okay, so it's going to give you 5.37 times 10 to the power of 14. Okay, and it didn't say a specific number of figures, so you could have just left it as 5.37 times 10 to the 14, or you could have left it with 5.4 times 10 to the power of 14 as well. Okay, so that's how you calculate, that's how you work out calculation questions with bacteria, basically, prokaryotic cell replication. Okay, you literally just use the formula and you work out the number of generations first, and then you plug it in to the formula to work out the final number of bacteria cells after a set time. Okay, so that's everything we're going to be looking at in terms of prokaryotic cell replication. What we're going to move on to now is viruses. Okay, so we did look at viruses briefly. We looked at the structure of viruses in a previous lesson when we looked at the structure of prokaryotes and viruses. But there's a bit more in terms of how they attack and infect cells. Now, viruses will come back later on in the A Level Biology specification. This is literally just the beginning of viruses. You learn about HIV as well, which is a specific virus, and how it sort of infects the body. So, so this is basically a part of the spec. So viruses are non-living, so they do not undergo cell division. So remember, they don't have any internal organelles. They don't have any internal subcellular structures. They are not classified as cells. If they're not classified as cells, then they cannot undergo cell division. Okay, they are non-living. Okay, they're really, really small particles. They are non-living. They can't undergo cell division. How they infect cells is inside viruses, they don't have any subcellular organelles or anything which takes up the inside of the cell itself. However, they do have some nucleic acid, okay, and it can be DNA or RNA within the virus. And what it does is that it attacks the normal body cell by injecting that nucleic acid um, into the normal body cell. So when you get a virus within you, that virus will take the nucleic acid from itself and it will inject it into the host cell. The host cell is our normal body cell, okay? And when it injects the body cell, the virus can enter the body cell, and what can happen is it can replicate inside the body cell and it can cause cell damage. Okay, and that's basically the only way that they can reproduce. If you've got one single virus particle, it won't really reproduce by itself. It can only reproduce once it's infected a body cell and entered the body cell. It will use the proteins that the body cell normally makes to replicate itself. That's how viruses replicate. Okay, they don't really they don't use any other means. They literally have to be present within your body. They will have to inject your body cell, and once they're inside, they will use that body cell as a means to replicate themselves. And when a body cell has too many virus particles within it, it will explode. And when that body cell explodes, that explosion of that body cell is what causes the damage. That's what causes you to feel unwell. Okay, so this over here is an image of a virus particle. We looked at this image previously when we looked at the structure. We're going to look at some exam questions which are related to the structure of the virus as well, because there's a lot of overlap between the ways viruses replicate, prokaryotic cell replication, and the structure. So just a quick recap behind it. If we look at this image over here, I know it's not the best image. However, we've got a virus particle over here. Now, in a virus particle, you have your reverse transcriptase, and the reverse transcriptase, um, the purpose behind the reverse transcriptase is to get the DNA copy and turn it into viral RNA. Okay, that's what it does. That's why it gets the um, body cells DNA and converts it into a viral copy of the RNA that the virus normally has. Okay, it has these attachment proteins. Now, if these attachment proteins weren't present, how is the virus going to enter the normal body cell? Okay, what's going to happen is that these attachment proteins, they're going to combine onto receptors which are present on the normal body cell, and when they attach onto those receptors, that's what's going to allow the virus to enter the cell. The receptor will be activated and will take in the virus. It will open the gate so the virus can enter the normal body cell. Now, we've got the viral RNA over here. So again, as I said, there's normally a form of nucleic acid that is present within the virus, and that's basically what's going to replicate every time a new virus is being formed. And then you have this lipid envelope. Now, not all viruses have this lipid envelope, okay? This lipid envelope only is present once body cells have been damaged. So let's imagine we've got our virus. Our virus enters our body cell, and then the virus starts replicating because it's using the proteins that our cell normally makes. It will start replicating itself. Now, in a body cell, there are too many virus particles now. Okay, now the body cell can no longer hold that many virus particles. So what's going to happen is that the body cell is going to explode. The body cell explodes, and obviously that's cell damage. Okay, all the organelles have been damaged, and it's literally ripped open the cell. However, there's loads of these virus particles, and remember, our cells have a cell membrane around them. This lipid envelope is basically that cell membrane that has been damaged. What viruses like to do is that once they've caused the cell damage, is they will take a component of the cell membrane with them. Okay, they will take a component of the cell membrane with them, just so because that cell membrane is part of your body, your immune system won't recognize the virus particle as being foreign. Okay, because it's carrying something that belongs within your body. Um, it's carrying a cell membrane that belongs in your body, that your blood, your immune system recognizes. So if it's carrying that cell membrane along with it, it's not going to be recognized as foreign, and an immune response is not going to be triggered. That's the purpose of the lipid envelope. And then finally, you've got your capsid. Now, this protein capsid is literally the protein coat of the virus, and it will protect that viral RNA that is sitting in the center of the virus particle. Okay, so that was just a little bit of a recap from what we have already looked at. in previous lessons. What we're going to do now is we're going to look at the process. Okay, now the process isn't that big and you don't need to know it in that much depth. When it comes to HIV, you will know that this will apply to that topic anyway. Okay, so it's just about getting an idea of what happens, and I've already been through most of it. So it says virus uses attachment proteins on its surface to bind to the complementary receptor proteins on the host cell surface. So over here, remember, if we have a look at this, we've got these attachment proteins over here. These attachment proteins, they're going to bind to receptors. They're going to bind to the receptors which are present on our normal body cell. Okay, so all our body cells have receptors. And because there are loads of viruses, that's why some viruses affect different parts of the body. The virus will bind onto the complementary receptor on the cell. Okay, now once it's bound to the complementary receptor, the body cell can take in the virus. And once it's taken in the virus into its cell, the virus can inject its DNA or RNA into the cell. So now, instead of a normal DNA within our cell, we now have viral DNA also present within the cell. Now what happens is that the host cell, the normal body cell, has nucleic acid, and that nucleic acid will produce the new viral particles for the virus. Okay, so that's basically what happens. Virus enters our body, finds complementary receptor on normal body cell, binds to that complementary receptor, injects its nucleic acid into the cell, and then the body cell's DNA will produce new viral particles for the virus itself. So more virus particles will keep replicating, so on, because the nucleic acid that's already present within the cell will do the work for you. Okay, and the process behind that we come on to later. The process behind reverse transcriptase and how reverse transcriptase converts the RNA into viral DNA is the process that we will come on to in a different topic.